This is episode 92 of the Immunology Podcast, Cell Death and Immunity with Dr. Pascal Meier. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, right after Halloween time, we have Dr. Pascal Meier from the Institute of Cancer Research on the podcast here to talk about his research on the complex relationship between cell death, spooky, immunity, and tumorigenesis. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
you know what? It's not spooky, Jason. It's actually pretty exciting. It's Immunology 2025, which is the world's leading annual all-immunology meeting. This upcoming year, so next year, 2025. It's in Honolulu, Hawaii. So join us from May 3 to 7, 2025 to connect, collaborate, advance your career, and access cutting-edge immunology research. Maybe hang out in the beach as you're on it. Abstract submission is open through November 19th. Learn more at immunology2025.ai.org.
I keep saying I'm super excited for that. Hawaii was my second home growing up, being from the West Coast, and my parents still live there part of the year, so I'm basically like a local.
You're a local.
Not on that island, but it's still not on Oahu, but still.
I've never been to that part of the Pacific, so I'm super excited.
I am excited to take you and give you the tour of
we got to start planning, Jason. We got to plan it.
Could you surf?
No, but I can learn. I mean, I can snowboard. So does that help me at all?
Yes.
Great.
It's just this very unique, like between the environment where it's located, just out in the islands there, it has a unique climate. And then like a combination of you're like in the tropics, but it's also still the US. It's it's kind of a unique overlap.
Be cool to go, do some, like nature expedition or something to see birds.
I think we should do a rainforest hike one day before or after. Maybe we'll do a luau too one night.
So if our listeners are also planning to go and don't have the advantage that I do of having, my personal tour guide, what should they be planning already? They're buying, they're getting their hotels and they're thinking about how long they should go for.
Book your hotel yesterday. You want to do a luau one evening. That's really fun and unique.
A luau.
Surfer, it's not actually in Honolulu, but there's the North Shore. There's really good Chinese and Japanese food on the main, especially in Honolulu. The beaches are all worth experiencing. You can do it local, even at Waikiki. It's high tourist, but you're local right there in Honolulu. God, I have to think about other Oahu. Each island has, is built on a volcano. And so there tends to be natural parks around every volcano that you can do some hiking at.
I can go visit volcanoes, see some lava.
Not on Oahu. You'd have to, if you want to go see live volcanoes, you'd have to jump over to the Big Island. Called Hawaii, Big Island, to see that going on. I wouldn't go, if you wanted to stay extra, I wouldn't go to Maui though, because the fire's there. They're still rebuilding.
Oh, yeah, that's true. Well, but we're not going to Oahu. That is in Honolulu. That's a difference.
Oahu was on the island of Oahu.
Oh, okay.
And
Honolulu?
Honolulu's fine. That's the city.
That's it's in the island of Oahu.
Right, exactly. That's the big city we're going to be at. And so you can check out the rest of Oahu. But if you want to see lava, that's the Big Island. I wouldn't go to Maui right now because of the fires. But there's all types of, beaches, and you can go snorkeling and scuba diving and everything.
No, that's not well.
It's standard tropical vacation, but
well, guys, if you're planning on going, better start preparing and start, thinking about all the fun stuff you can do before. Maybe you can go. I'm planning on being there before because I need to get over the massive time zone difference there's going to be. It's literally like on the other side of the globe. So I'm like mentally preparing. It's going to be worse than the time I went to Vancouver. It's going to be tough. So,
it's like half a day apart, right?
Yeah, pretty much.
Oh, man.
But well, all right. But, let's think about the immediate and stop daydreaming about our future in the beach.
Let's talk about the beginning, Brenda. Let's talk about the beginning and not the future. And let's talk about stem cells.
Ooh, how poetic. The beginning of you, of me, of everyone. Okay, what's up with the stem cell?
All right. So I like the merger. It's a little bit of stem cell podcast and an immunology podcast, but still very immunology.
Daily in our room, we're coming for you. No, I'm just kidding. That's the host of our sister podcast, the Stem Cell Podcast. If you haven't listened to it and like stem cells, you should also check them out.
But you know what has stem cells? The hematopoietic stem cell compartment. All right.
Right.
It's in Nature. Came out. So received the 4th of December 23. Accepted the 18th of September 24. Ouch.
Oh, less than a year.
Published online the 23rd of October. Title: In Nature, Differentiation Fate of a Stem-like CD4 T cell Controls Immunity in Cancer. First author, Maria A. Cardenas. Corresponding author and last author is Hayden T. Kissick. All right. So they were trying to understand what's going on with tumor-specific CD4 cells and understanding the differentiation fate of them. It's not entirely clear how they got to wanting to study this. I'm sure they had preliminary data from something else. They started looking at single-cell RNA sequencing, they said, on PD-1 positive, CD45 negative, CD4 T cells infiltrating kidney tumors. How they got to that, I don't know, but that's where the story starts today. So what they found, and this is what's really fascinating, is there are these stem-like or stem cell capacity CD4 cells adjacent to the tumor that are, and I'll get into the markers in a second, but at a high level, they primarily reside in the tumor draining lymph nodes, and they're restricted in their capacity to expand by Tregs. If you remove Tregs by depletion, you get differentiation into Th1 cells. And then the Th1 cells, by interferon production, induce robust effector differentiation for TCF1 positive CD8 cells, which then controls cancer. If you enforce T-bet expression, so you overdrive T-bet in these stem cells, you can overcome the Treg suppression effect. And so that can restore it even if you have the Tregs there. So, and then they find, if they look in cancer patients, high level, right, that they see that people who have more of these cells do better, especially if they have high T-bet with these cells. So if they have more T-bet high of these stem cells, you get more Th1 cells and more CD8 cells, and they live longer. So they just look post hoc at outcomes in people. So they do a lot of sequencing. They identify these cells are TCF1 positive and Lin negative. Most of this approach is around single cell RNA-seq. And then they do some follow-up work. But mostly it's they take these cells that from patients to identify it, and then they can amplify and trace and do a little bit of signaling as well. And then they also then recapitulate this in a mouse system as well to see that that's there. And then they use like smarter cells, these cells that have an LCMV virus. If I may be having the wrong virus in my brain, but I think it's LCMV virus. And they can use that as a tracer since it's antigen positive and track the ability of these cells to expand and kill that virus as well. And so what they see is that at baseline, the Tregs are holding them in check. If you deplete the Tregs, the anti-Treg antibody with a FOXP3 knockout, pick something, right? You get expansion, but you can also just overdrive T-bet and you get this. And they show that it's a clonal expansion. They show that, stem cells that have these pseudo stem cell like ones that are targeted towards the antigen are the ones that then expand. They're not resident T cells. They do some pretty sophisticated depletion where they'll deplete the entire host's circulating cells but leave their resident cells, right? Because they last so much longer when you do a transplant. And so they do that time depletion study. When you do the depletion, it goes away. So it's the ones that are draining in the lymph nodes that do this, not just resident T cells, right? And they can dissect out what they're seeing and get there. And so the paper goes into a lot of detail. I think it hits that nature level of being a pretty big deal in terms of identifying a regulatory pathway of Tregs controlling this population, getting rid of Tregs or overdriving the population to shift despite it has therapeutic benefit. And then the paper is very bulky in terms of all the mechanisms and tools they use to show this. They don't just show this one way, right? They use specific tumor models that are targeted by T cells that they add in and can then trace. So they do a lot of lineage tracing. They use SMARTA cells. They use a significant variety of techniques. They map out the transcription factors involved. They identify that these CD4 Th1 cells produce IL-1. IL-1 induces CD8 activation. So if you kill IL-1, or sorry, not IL-1, interferon, it works. Right. And so they actually trace it step by step all the way through. And it's pretty cool, right, to know that there is a resident population of stem cell like cells, down the path, but still stem like that were ready to differentiate or being held in check by the Tregs.
That's a really nice example of, this dominant suppression that Tregs can do. I wonder, is there any experiment in which if you have a situation in which you deplete Tregs and then you initiate this differentiation, then if you put Tregs back, does this fix it?
It depends on timing, right? So if you already let them differentiate and start doing their thing, then you have a kill. They did look at the timing of it, right? So if you deplete Tregs, throw them in, and then stop the depletion, it's too late. If you let the Tregs recover as if you didn't deplete, then then the Tregs hold them in check. Or you can have it in place and then deplete Tregs, and then population will pop up and kill the cancer.
Right. I'm also thinking on the other side in the context of, this is in the context of cancer, in which you don't want the Tregs, but I wonder in the context of autoimmunity in which you do want the Tregs, and whether there are Tregs, whether there's a comparable situation in an autoimmune context in which the presence of Tregs.
I'm sure of it, but they don't get there, although you can kind of see that with their traces with the SMARTA mice.
Mm-hmm. Yeah.
Cells. That it is holding antigen-driven attack and presence.
And so in terms of like therapeutic, what we learn that would affect therapy, what do they suggest? How can you apply this?
You could either, so they don't get there in therapy, right? But how I would do it is you could do a Treg depletion acutely, right? You don't need it forever.
Right.
Treg with an antibody, boom, these come back. There you go.
Yeah. Yeah, and that would be enough to kind of tilt the balance.
Right, exactly.
Right.
Activating T-bet seems a lot harder, frankly. Yeah. I don't like trying to turn transcription factors on.
Yeah, that feels a little bit more complicated, doesn't it? Okay, so I will also continue talking about Tregs. What a coincidence. But in my case, I will be talking about Tregs for autoimmunity. So Tregs as the good guys, the guys that we want to use. This paper I will discuss today is titled Human OX40L CAR-Treg target activated antigen presenting cells and controlled T cell alloreactivity. And it was published in Science Translational Medicine on the 16th of October. And the first author are Jianliang Rui from corresponding authors Victor Tachev and Leslie Keane at Harvard Children's Hospital, I think. And I like this paper because it's interesting. I don't want to say unconventional or unorthodox because it's not that strange, but it is a slightly different approach to using chimeric antigen receptor constructs for therapy. On the one hand, of course, you're not using it to direct the activity of a cytotoxic T cell or an anti-cancer T cell. In this case, you want to direct the activity of regulatory T cells because, of course, as their name suggests, they are capable of, and as your paper also indicated, their presence is very important to prevent the immune responses. And in this case, we're talking about, unwanted autoimmunity, or in particular, graft versus host responses, in which if a patient, for example, has leukemia and needs to receive a bone marrow transplant, that usually is from another person, usually, like pretty much always is from another person. And there is the risk that that bone marrow comes with T cells that are capable of recognizing something on the recipient's tissues and the recipient's antigens and generate what is known as an allo response. And this is a major risk of bone marrow transplant. And it's difficult because on the one hand, you could potentially deplete all T cells really, really hard, like really try very hard to remove every single T cell. But if you're getting a bone marrow transplant because you had leukemia, those T cells can also sometimes it's good to have some T cells, some effector cells coming from your donor, because they also carry on this graft versus leukemia or graft versus tumor response, because otherwise you are left without any T cells for a substantial amount of time. So that might actually be worse. That the alternative. So usually you still get some T cells, and this can result in graft versus host disease. And so one way, people are treated with corticoids or corticosteroids or other kind of immune depressors, and that kind of usually helps. So that can reduce the risk of graft versus host disease. But sometimes you also have like long-term or chronic situations. So it's really hard to treat. Nowadays, we only treat the symptoms. We only treat like down once the response exists, and then we just try to dampen as much as we can with immunosuppressants, which is also, of course, not great because you do need your immune system. So that's always a problem. So what to do? One of the things that you can do is try to harness regulatory T cells, which are, by definition, designed to specifically prevent the activation of the immune system in the wrong place. And if you direct them to the right thing, you can maybe generate local suppressive responses, and that could keep the graft versus host disease in check. And this is kind of the idea they want. So always what is hard, if you have a CAR, that has an antibody, the domain of an antibody, so you're directing against some kind of receptor, some kind of protein, surface protein. Always the question is, what do you direct it to? And in this case, what they suggest is, why don't you direct the Tregs against antigen-presenting cells that are activated and that are presumably, inducing this immune response. And this is where OX40 ligand comes into play, because this is a molecule that gets upregulated in inactivated antigen-presenting cells, and it interacts with the receptor OX40 on the surface of T cells, also known as CD134, and this is a co-stimulatory receptor. And this interaction between OX40 and its ligand seems to really has been associated with the pathology or with the immune response in allo immune responses. So that is kind of the reason, the background behind this decision. So what they do, basically they generate a CAR that has that is expressing OX40 ligand, and it's driven. So OX40 ligand specific CAR, so an antibody against is based from an antibody binding to OX40 ligand, and that is expressed under a FOXP3 promoter. So the idea is in this way, you're only going to be expressing this on proper regulatory T cells. And they generate this construct, and basically they show to see how does this affect, how does these Tregs interact with antigen presenting. And it's very interesting because they do show that indeed Tregs demonstrate they get activated when they're co-cultured with OX40 ligand positive cells, and they get activated. They show upregulation of Treg activation markers that are associated with Treg activation, and in this case they are talking about CD69 and CD71, which are not the most common, but then they also are talking about activation markers or like molecules that are part of the suppressive phenotype of Tregs. So things like CTLA4, GARP, LAP, they are all part of the kind of the Treg phenotype. And they show that these cells can act as Tregs upon stimulation through their CAR. And this can functionally suppress another reactive T cell proliferation. They have proliferation assays, and they show that they can prevent the activation of conventional cells, which is, the whole point. They do what is known as mixed lymphocyte reactions, in which you mix antigen-presenting cells from a different donor. So you have this mismatch, and that's what the T cells recognize. So if you just put, PBMCs from one donor with T cells from another donor, you're going to have some T cells are going to start growing out, and this is kind of a model of an allogeneic response like GVHD. And this is something they show that they can prevent by using this Tregs. And I guess the important thing is that in this way, these Tregs are better than Tregs that don't have this CAR. They do this in a much stronger manner. And what is interesting is that they go a little bit into the mechanism. So one of the things that we know is that Tregs are little gluttons. I think that's the word. They are capable. It has already been described that when they bind to certain of the receptors, allows them to really trogocytose. That is, remove whole pieces of membrane from the cells they interact with. And it has been, suggested and shown that through this mechanism, they can do like deplete cells of antigen-presenting cells of certain molecules such as CD80, CD86, MHC class 2, and also OX40 ligand. And basically they compare and they look at whether these Tregs are, removing more OX40 ligand from the APCs, and they show that the addition of this CAR increases the capacity of the Tregs to remove OX40 ligand from the surface of antigen-presenting cells, which presumably makes them worse antigen-presenting cells. And that's kind of the major way of the mechanism by which these Tregs are superior to the Tregs not expressing this CAR. And they have, say, in vivo model, they have a xenograft model in NSG mice. Basically, they put PBMCs so in NSG mice, and then they put the CAR-Tregs, and they show again that they work, and that they still keep this important graft versus leukemia effect. They have also some experiments which they add leukemia cells, and they show that they can still, that this suppression of the GVHD does not necessarily mean you're suppressing the response against the tumor cells. So I thought it was very cool. I think this is the first time I hear of this approach. And it's nice because then in principle, you can make this CAR-Tregs would be useful for various diseases as long as you have, this OX40 ligand component, as important for the activation of the immune response. So that's cool. That's nice. That's a nice read.
I like it. It feels like the right amount of Treg.
The little robots keeping the peace.
All right. Well, I have another one for you, also from Nature. I have no good segue here. It's about chromatin remodeling and heart failure in immune cells. And the title is Chromatin Remodeling Driving Immune Cell Fibroblast Communication and Heart Failure. So first authors are Michael Alexandrian and Arun Padmanabhan. And last author is Deepak Srivastava. All right. So complexity here. So real quick. So it came out December 23rd. It was received December 21, 2022. It was accepted 19 September 24 and published 23rd of October 24.
Oh my God.
So the high level story is pretty cool. I don't know why it's in Nature versus like a subjournal like Circulation or Heart or something, because it's cool, but I'm not sure how broadly like broad it is, right? And this may be just like my knowledge gap that I'm not quite sure on of types of things, but like it's still cool overall. So high level. We know that inflammation after heart damage and other situations that lead to worse outcomes because of fibroblast activation and a combination of scarring and improper wound healing and fibroblasts being bad. Okay. And there's nice mouse models where you can ligate stuff off and jump up the blood pressure on one side of the house to induce on the heart to induce heart failure. And that's the model they use here. And then they can study what happens afterwards. So what they show is that these CX3CR1 macrophages drive disease state at baseline. Because what they show is normally when these macrophages, I mean, there's going to be a lot of letters here. So when these macrophages, these CX3CR1 macrophages, when these macrophages are in the presence of a heart failure situation, there's a transcriptional coactivator called BRD4 that is there, and they see down to the chromatin level that is a large enhancer of IL-1 beta. And IL-1 beta secreted activates REL-A, also known as P65, near the transcription factor MEOX1, resulting in a profibrotic response in human cardiac fibroblasts. And this causes heart failure in the models. So at every step, you knock out BRD4, which is what they did first, myces don't get heart failure. You do targeted IL-1 beta depletion in the macrophages, or you do systemic IL-1 beta inhibition, you don't get the heart failure. So that's the signaling high level. Like, okay, that's a pretty cool story, right? The macrophages release the IL-1, and the IL-1 really makes this program that's profibrotic off the MEOX1 transcription factor and go live, and it's all mediated by IL-1. But what they do that's really cool is they look at single nucleus RNA-seq and single cell RNA-seq. They get down and attack-seq, and they get down to the fact that it's the BRD4 coactivator binds to the super promoter of IL-1, and they really map out everything down to the spot on the chromatin where this action is happening for every one of these genes along the way. So this is where I don't know if that's super cool or not super cool to know that. I think it's neat to know. It's a lot of detail I don't usually get to see. So that's really awesome. But high level, macrophages secrete IL-1 during heart failure, which makes fibroblasts become profibrotic and cause the heart failure phenotype as opposed to the better phenotypes. And if you block it at any step along the way, you stop the process. And now, so the high level is very quick. And then this paper has a lot of detail mapping out this spot on the chromatin and everything else and doing all the knockouts and conditional steps to show that each step is necessary and sufficient for the next step to occur all the way down the cascade.
Bad macrophages. Always messing around.
It's not like those T-regs, right? That, they don't do anything.
No, they do good things. They are pacifists. Sometimes they just are in the wrong place, but their heart is always in the right place. So does this mean that we should be like injecting anti-IL-1 to like people that had a stroke, had a heart attack or something? Is that what they are proposing?
Yeah, essentially like during heart attack, when you're in the recovery phase, you should get some IL-1 beta antagonist, maybe.
Do people with a heart attack receive any kind of immunomodulation?
No.
Like after, no, right?
Aspirin.
Aspirin. Yeah. Well, that's because I heard a lot about in other contexts, like damage to the heart muscle. When you have a stroke in the heart muscle, you have like a
If you have a scar, it doesn't work as well.
yeah, and that, for example, yeah, it's because of the inflammation that is generated, and uh, that damages the muscle fibers themselves.
There's that too, and then inflammatory healing is less good than non-inflammatory healing,
right?
because of the insults. You get inflammatory healing.
You know who can keep in check the inflammatory healing?
Tregs.
Indeed.
Yeah, I mean, if we just had or just awash in Tregs, I'm sure none of us would have cancer either.
None of us would have cancer. Are you
a little bit?
I'm not sure. I looked at him in the video, dear listeners. I don't know if he's being serious or if he's pulling my leg. Let's finish up our discussion for today. I guess it's still about cancer. So my second paper is about cancer. And now that I think about it, I think this paper is from the same authors from the same group that you talked about last time. Like, I know this name. It sounds very familiar. So the paper is called Programmable Bacteria Synergized with PD-1 Blockade to Overcome Cancer Cell Intrinsic Immune Resistance Mechanisms. Oof, that was a mouthful. First author is Fang Da Li, and the corresponding author is Nicolas Arpaia at Columbia University in New York. And I think that's the same author from your paper about probiotic, neoantigen, I forgot. So they seem to really like doing stuff with bacteria and just putting it into mice and see what happens. So let's jump in. I think this is really cool. This is like synthetic biology, and a very nice application. I think it's pretty elegant. So I like this. So the idea is, what is the problem here, right? You have tumors. Tumors are bad. Tumors, when they grow out, they make this microenvironment. T cells can't live in this microenvironment. This microenvironment is depleted of nutrients. And yeah, they're just not happy. And it's also depleted of kind of like signals for the T cells to keep going. And one of the signals that seems to really help T cells get activated, and your macrophages and other immune cells get activated against the tumor is interferon gamma. So we know that if we could magically put interferon gamma inside the tumor, that usually helps the cells in the tumor get activated and get the right phenotype and fight the tumor. But that's kind of hard to do. Interferon gamma is notoriously dangerous systemically, so you cannot really inject like intravenously, hope it makes it to the tumor at concentrations reasonably enough without killing the patient, basically. So that's really tough to get interferon gamma, administer interferon gamma without massive side effects. So, as the saying goes, if you need, if you need to get to the mountain and bring the mountain to you in a way, so you need to bring the source of interferon somewhere inside the tumor, so it stays there. It has this topical, this concentrated effect, but then it's not systemically everywhere. And I like this because then they thought, well, let's make bacteria do that. So they designed this bacteria, which they call slick interferon gamma. And this slick design is basically uses a probiotics or non toxic E. coli strain, and they engineer it with a circuit that is very interesting because it's a quorum sensing mechanism. So I think they might have used the same for the paper you discussed. So basically, this bacteria, they have a promoter that controls the production of a single signaling molecule called N-acetylhomoserine lactone, AHL. And as this bacteria grow and they accumulate, the density, so the amount of this molecule in the environment increases. And when certain critical bacterial population is reached, and this results in a critical concentration of this AHL, this activates a phage-derived lysis gene that then causes most of the bacteria to die. And because in this plasmid, there's also interferon gamma, which is produced but not secreted when the bacteria dies, this interferon gamma gets released. And then, but this doesn't kill every single bacteria. And also you have a few bacteria that survive, and then, this quorum sensing is gone because most of the bacteria are dead. So they start growing again. They reach this critical mass. They die. They release interferon gamma, and they just keep doing that forever. I didn't quite understand if they just do it forever or like at some point they would be cleared by the immune response. I'm not sure, but basically you have this certain amount of cycles of it, and then you have wherever the bacteria are growing, which is not everywhere. So I think that's the point. They grow mostly inside the tumor, where the conditions are kind of better for them, I guess. And that's where they're going to be releasing these pulses of interferon gamma. And so it's really cool. It's like, little, self-releasing, self-multiplying pill that can find its way to the tumor. Or, the first experiments, they actually just like inject it inside the tumor. They start doing, so they have some in vitro, first they do in vitro studies, and they show that as they grow, they can activate macrophages by the production of interferon gamma. And then they have, mouse models with MC38, colorectal cancer line. And again, they show that by doing this intratumoral injection, they can really reduce tumor progression without systemic toxicity. And this is the main advantage over regular interferon gamma injections. And they show that, when they do this, what happens is that these bacteria, they drive expansion and activation of tumor infiltrating cells, CD4, CD8 cells, and K cells, and basically it becomes a more pro-inflammatory environment. And this is like what's driving the tumor rejection. And that you can even have what is known as this abscopal effect, in which if you inject, you have two tumors in different places, injecting one tumor allows the response to be kind of transmitted to the other tumor, even though that tumor did not receive bacteria. And so this is really nice. So basically shows that this is a better way of administering interferon gamma to the tumor. They also eventually show that you can even put this bacteria IV, and they find their way to the tumor, and they are still working without toxicities, without severe toxicities. And what is interesting is that they did all these experiments using this this overexpressing MC38, but then they go into MC38 without OVA, which is very, very immunogenic. So that even with this system, MC38, no OVA, still kind of escapes. They see a reduction in the growth, but still eventually it escapes. So what they do is they combine the treatment with the bacteria, with the slick bacteria, plus PD-1. So they inject the bacteria first, and then they wait for a little while, and then they put PD-1 treatment. And this is because one of the things that they observe is that there's upregulation of PD-L1 in the microenvironment. So they say, well, these T cells are being, really inhibited. So the combination of both really, again, controls the tumor growth. And it's better than, either PD-1 alone or the slick cells alone. They also say what happens, I think it's also interesting, is that they look at the situation, which is very common, which tumor cells lose expression of MHC class 1. So then they're going to present antigens again anymore. So then T cells, what are T cells supposed to recognize if there's no MHC class 1? They have a model in which they knock out beta-2M, which is important for the kind of the assembly of the MHC class 1 molecule. And they show that they can still trigger cell death. They can still, with this slick interferon gamma E. coli, they can still mediate tumor control. And that now they do some studies, and they conclude that it is NK cells that are taking over when there's no MHC class 1, because loss of MHC class 1 becomes a it's a loss of negative signal to the NK. So all of a sudden, this missing self is detected by the NK cells, and interferon gamma also activates NK cells, so they are also kind of licensed to kill the tumors. And they also look into, what happens if you have less sensitivity to interferon gamma. So they show that even when situations like JAK1 or JAK2 knockout or like loss of this expression, which is also a very common mechanism of immune escape, can still have significant anti-tumor activity, even with a conventional interferon gamma response in the tumor cells themselves, which to some extent, sometimes a lot of interferon can even kill the tumor cells directly through this JAK1, JAK2 signaling. So overall, they talking about little biological robots doing the work for you. They show that if you can bring the interferon to the tumor using this kind of harmless bacteria that have this forum sensing and they have these pulses of interferon release, that it can really remodel the tumor microenvironment and allow the T cells to be activated and functional. And if you combine that with checkpoint inhibition, anti-PD-1 therapy, they really synergize. So I think it's super cool.
Yeah, my only flag is I don't understand how they have no tox. If they can cause just any cell that uptakes these E. coli that's an immune cell, can cause piles of interferon gamma. Oh, there's no toxicity here. I'm like, huh? Did they inject it in healthy mice that had no tumors or anything and show that the mice are perfectly fine with no autoimmunity? Or like, hey, on top of the terrible cancer the mice have, we don't see anything else, which is not the same.
Yeah. I'm not sure they have that.
That was my
and
big
also
question.
and also all of these assays are like a month or so. So I'm not sure what happens after. I mean, they might still have to get
if you provide interferon plasmids that E. coli can switch back and forth, you can make your entire gut develop into E. coli interferon producing microbes just by bacterial sex.
Yeah.
And like, I have questions. It's cool, but I have lots of questions on human application.
I think you have a really good point. I also thought that's why I think, there's a lot of holes in the argument, so to say, or I think the principle is really cool, but you really need to make it watertight, and take care of all these necessary potential side effects that you really don't want, but they're kind of inevitable if you don't know anything about it.
Yep.
But it's fun. It's a fun idea.
All right. Well, we got to move it on. And we're going to be speaking to Dr. Pascal Meier at the Institute of Cancer Research in just a moment. But before we get to that, ensure reliable results with your immunology research, from primary human cells to cell isolation kits, culture media, supplements, and antibodies. STEMCELL Technology provides the tools you need for every step of immunology research. Interested in cell isolation? Use EasyCep to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Cells are viable, functional, and immediately ready for your downstream applications. Learn more at easycep.com.
So now for the second part of our episode, we are very happy to welcome Dr. Pascal Meier. He is the group leader at the Institute of Cancer Research in London. And he's going to be talking to us about cell death, immunogenic cell death, and maybe some unexpected correlations there. Thank you, Professor Meier, so much for joining us for the Immunology Podcast.
Thank you, Brenda and Jason, for having me on the show. Thanks.
Thank you for being here. All right, Brenda, do you want to go first on this one?
Yeah, let's talk about death and why. I guess there's not every death is the same. And I think that's a lot of what your lab works on different types of cells die under in several circumstances, but there's some types of demises that are better when you're thinking about an immunology response, for example, in cancer settings or maybe in other circumstances. So maybe we could start about talking about cell death? What does it mean? And what are the different types that we kind of study, or you in your lab and your group is thinking about?
Yeah, so this is really an essential topic that fits into many, many different aspects, from tissue homeostasis, but also for mostly host pathogen interactions. And their death, I think the one thing that I would like you to take home from this podcast really is that death is not an endpoint. It's in fact the beginning. It's the beginning from the communication of the cell that dies with the tissue microenvironment and the immune system in particular. So when a cell dies, it really communicates itself to the neighbors. And in most settings, this is relatively benign. And there's a good reason for it. I mean, we clear about 200 billion cells every day for good reasons. You don't need them, you turn them over. And of course, the immune system shouldn't be sidetracked by this. But then there's other death modalities that are, for example, lytic, necroptosis, and pyroptosis. And there, the event really is geared towards alerting danger, alerting the immune system. And it has now become clear that the different types of death modalities that have evolved have really sort of geared towards this type of communication. So death, in that respect, is really not the end. You need to know why a cell has died. And it's important to remember, no cell dies in isolation.
So do you want to cover, I guess, before we go into what happens after you die in more detail, you mentioned that there's types of death. So could you cover the main types of death that we should be considering here before we go into a discussion of what happens with those deaths?
So, I mean, you guys as an immunologist, you care about death because it's the beginning of the immune response, right? You need death for cargo uptake, for sampling, epitopes, foreign epitopes, or tumor-associated neoepitopes. But just a mere uptake of a dead cell by a antigen-presenting cell is not sufficient. It's required, but in fact, if a dendritic cell eats an apoptotic cell, it actually shuts itself down. It prevents its ability to maturate into a DC that can efficiently cross prime. So what you also need is the adjuvancy. And here, when I refer to these lytic forms of death, it's not actually the fact that you blow up that is immunogenic. So the machinery that actually triggers this blow up, at the same time as it builds the machine that kills the cell, it also drives production of alarmins. So that's the transcriptional response. So activation of NF-κB and interferon drives this synthesis of alarmins, which then are most effectively released when the cell blows up. Like, for example, IL-33 doesn't have a signal peptide. Also, IL-1 beta doesn't have a signal peptide. They don't come out of living cells. They come out of dead cells that have blown up. So in this respect, so necroptosis and pyroptosis, they have evolved in providing two signals. One is a dead cell, and the second one is the adjuvant. And apoptotic cells do not do that. So for a start, you retain your membrane permeability, and also caspases, they actively incapacitate the production of these dangerous signals. They cleave, for example, C-gas, they cleave IRF, and that prevents the production of interferon and the production of alarmins.
So would it be fair then to say everything but apoptosis is immunogenic largely? So necroptosis, pyroptosis, necrosis. I know apoptosis can be, but would that be kind of the main line of where apoptosis is more an end to an end and everything else is the beginning of the next step?
So unfortunately, it's not so simple because also apoptosis can be immunogenic if you provide the right adjuvancy either from nearby or if you derail the apoptotic process. There's also, of course, you can have apoptosis together with NF-kappa B or C-gas activation. This can happen. It all depends then also on the kinetics of apoptosis. If you have a huge amount of apoptotic cells that cells cannot gobble up, then you also have secondary necrosis that can contribute to an inflammatory condition. Of course, that itself has a secondary consequence through macrophages and other immune cells. So it's not as simple as that, but in general, apoptosis, it tends to be immunologically silent. It's not silent with regards to information, but the information that is released by apoptotic cell is more geared towards cell proliferation, towards a wound healing process.
So if we think a little bit about the mechanisms behind like more inflammatory cell death, like more pro-immunogenic cell death. So some of your work has looked into RIPK1 is a kinase that is very well associated with this process. So maybe as an example of like the pathways, like the signaling, how exactly do we understand the difference, and how do these cells transmit this information if it has to be an immune response? Do you want to maybe comment on that, on the signaling around an immunogenic and a non-immunogenic cell death?
Yeah, so RIP is a major stress. RIP kinase 1 is a major stress sentinel. It operates downstream of many pattern recognition receptors, but also death receptors, including TNF receptor 1. And it functions as an adapter. In most settings, it is benign. It helps to activate NF-kappa B and chunk signaling, and nothing happens. I mean, most cells are super happy to experience TNF. But what TNF really does is it probes the system, it probes cells for fitness. And if everything is fine, then this RIP1 gets nicely ubiquilated, it gets recruited to TNF receptor, it builds a ubiquitin-dependent signaling complex that is geared for survival and mainly drives activation of NF-kappa B, an inflammatory response that is timed. But when the cell is under stress, that means, for example, is a pathogen is on board that interferes with IKK, with TAC, to prevent an inflammatory setting, then RIP1 turns from a survival molecule, pro-survival molecule, into a death molecule. Because under these conditions, it's better to throw away that cell and start again. It's also important to remember, I mean, our body doesn't have a problem with death. We can always make more, but we have a problem with pathogens. And that's really where these different death modalities have evolved out of. It's a rat race between us and pathogens. And therefore also in different species, you have these pathways slightly different. For example, this lytic form of necroptosis and paroptosis, they do not exist in carnivores. They have lost them. Whereas we have it, mice have it, many other species have it, bats have it, but carnivores not. They've lost both paroptosis and necroptosis. Most likely because the pathogens are such that either they have won or it would not be beneficial for the carnivores because of what they eat.
That's very interesting. I was not aware of that. Yeah, I guess it also cell death, when you think you say our bodies are used to it, and it's such a important, especially I'm not a developmental biologist, but it's obvious that through development of multicellular organisms, as you are growing, as you're changing your structure, you're going to have some cells are going to become redundant. So these pathways must be so conserved to some extent within different organisms of different branches I guess most animals have a lot in common in these pathways. And I guess does it extend also to other, like, do plants have similar pathway? I don't know, maybe it's outside of your scope, but uh, what do we know about other groups of organisms dealing with cell death?
No, I mean, death is important in many organisms, even bacteria, because you want to you want to preserve your friends. And the easiest way, of course, to limit pathogen propagation is to burn down the house. And this is a common principle. Of course, if you are infected, then you stop the propagation by killing the cell. That helps in many settings. So death is easy to come by, but it's also regulated quite complicated in a way. So you have backup system. For example, necroptosis doesn't happen normally because apoptosis also caspases block necroptosis, and it only steps in when apoptosis, so the primary cell death pathway, is actually blocked. So you have this booby trap that opens when you interfere with the normal pathway. So these are hooked up, and actually, if you, for example, have a cell that would die by apoptosis and you block apoptosis, then it goes into necroptosis. And if you also block necroptosis, then actually it switches into pyroptosis. So there's different booby traps, because once the cell realizes it needs to die, it will find a way to do so.
So to continue on this, though, how about, we talk about death as normal. It's a good mechanism. We have some booby traps. How about when it goes wrong?
Yeah, playing with fire can be dangerous. And of course, it's now becoming increasingly clear that chronic death activation, when these tight control mechanisms around RIB1, for example, don't play properly, then you come down with Crohn's disease, arthritis, neuroinflammation. And it's now clear, at least from the murine model systems, that many of these chronic inflammatory diseases, they're not due to aberrant inflammatory signaling. They're due to aberrant cell death. So if you block cell death, you prevent many of these chronic inflammatory settings. Of course, yes, if you start constantly driving in sort of immune call-out alarm bell, then that causes real problems.
So I want to bring kind of a, I don't want to say a buzzword, but something that also has become very intense area of study lately. Is there some connection? What is the relationship between cell death and senescence, for example? Is it something that so people study a lot of senescence and cells, in a way, cells that won't die. So what is going on there? Is it something that you kind of think about as well?
Yes, I do think about it, but we not actively work on this topic. So this has particularly hit the limelight recently, also in the cell death field, because it becomes clear that cells that undergo minority MOMP, so MOMP is the mitochondrial outer membrane permeability, so they then leak out mitochondrial DNA, which activates C-gas and STING, and that causes this SASP, this senescence-associated secretory phenotype. And so it's partly regulated by the BCL2 family members, so because you leak there to some extent, you have this minority MOMP that contributes to this, and therefore also, not surprisingly, the so-called senolytic are nothing else than BCL2 inhibitors. So what are you actually doing? You're allowing these half-dead cells to actually die completely. So frequently these senescent cells, these are aberrant cells that are sort of half-dead or semi-dead. They have
Zombie cells.
Zombie cells, exactly. And then of course, you activate responses to these events, which in turn derails the cell.
Right. Is it correct to assume, because I had the impression that usually kind of permeabilization of the mitochondrial membrane usually triggers cell death. But you're saying that BCL signaling is preventing that connection.
So I mean, that is true. The point of no return of a cell is really MOMP, so the total breach of the mitochondrial outer membrane. But not all of your mitochondria need to undergo MOMP, and if you have a good proportion of mitochondria still intact, you can function. And there's ways that you can also prevent the actual execution of death by caspases. So caspases, in that respect, they can contribute to the death, but caspases per se are actually not determinant of cell death. So this is also a bit wrong in the literature. Caspases contribute to the kinetics of death, but they don't determine whether you live or die. What they do really, or what they are important for, is they modulate how death is perceived. So, for example, in apoptosis, they speed up the death. They contribute to the secretion of eat me signals and find me signals, and they incapacitate more immunogenic signals. So in that respect, caspases should be really considered a sort of modulator of death perception, because the point of no return resides with MOMP.
So are you then, kind of quick follow up on that, people that do all this caspase, cleavage, Western blots, what are your thoughts? Should people stop doing that and saying this is cell death, and then look into mitochondrial potential or something like that?
Yes, absolutely. I mean, in that respect, of course, I mean, you can ultimately also, I mean, it's no assay on its own is sufficient. You always need to look at different ways. So if you see caspase activation, not necessarily that means the cell is dying. That there's a lot of caspase activation required for signaling. There's sublethal caspase activation that can contribute. And you can block caspase activation, the cell still dies. It just takes a bit longer, and the death is a bit more messy. In fact, if you do that, you convert an apoptotic immunologically silent death into an immunogenic version of death. And this is also now in a therapeutic setting, an idea that actually you want to prevent caspase activation. And that would be beneficial for an anti-tumor immune response.
That's exactly what I wanted to go into, which is, can you talk a little bit about the therapeutic angle? So, I give you some chemo, I kill your tumor cells, they undergo not just apoptosis, but all the osis. And life is great, right? Because we now have immune activation. And so all our first line chemos from the 60s all worked perfectly and cured cancer, but they didn't. So tell me more about death as a therapeutic and what you're looking into there.
So, I mean, it's rather unfortunate that over the last 40 years, we've developed all these chemotherapy, radiotherapy, and all these other approaches. They all trigger apoptosis. So really, we need to get away from this. We need to try to actually prevent caspase activation, prevent apoptosis. This is much more immunogenic. And in a way, it's not surprising, right? I mean, what you need to do, you need to trigger an event that looks like a pathogen infection. Viral mimicry is here the buzzword. And you can do that by blocking caspases and by activating, for example, pattern recognition receptors, or converting a TNF response into a RIP-mediated necroptotic response. And we and others have data that clearly indicate if you switch apoptosis into necroptosis, this is much more efficient in driving an anti-tumor immune response. And you drive not only activation of CD8 positive T cells, you also trigger immune memory.
And this is because the caspases don't cleave everything up?
So the precise reason is not entirely clear. It's early days. It's more the thought that you actually convert the death into a more professional activator of the immune system. So caspases, yes, they are effective in preventing these events, in preventing immunogenic signal. Apart from if you look at caspase 1 and caspase 10, of course, these are professional alarming producing cells. But caspase 3 and 7, they do just the opposite. They dampen down the signal, and you don't want that.
So from maybe again, it's a bit too down the road, but so when you're thinking of this kind of therapies that are enhancing the immunogenicity of the cell death, they are diverting away from apoptosis. So what are the immune cells that are responding to this? This is the macrophages and dendritic cells. Which are the signatures that become the most clearly benefited by this kind of treatment?
So we certainly we are still in the midst of that. So the entire field of cell death is looking into that, which immune cells actually are most responsive. And of course, dendritic cells sit at the heart of that because they gobble up, they activate ultimately the T cells. But it's most likely a multi-prong approach, and macrophages will be important too because they are super sensitive to die by necroptosis, much more sensitive than dying by apoptosis. So they're geared towards more the necroptotic signaling arm, because ultimately the danger that we are exposed to are pathogens all the time. So we're here not to survive cancer, we are here to survive pathogens. So what we need to really try to do is to change our therapeutic approaches into these host pathogen scenarios. And Jason, of course, you're here at the heart also with looking at the microbiome. Of course, that also means that patients will respond differently depending on the different microbiome they will have, because your immune system is less or more trigger happy depending on your microbiome.
Yes, very, very true. So it sounds then therapeutically you want to maybe use the chemotherapy we have, but add a caspase inhibitor or something else to shunt it to a necroptosis. So it becomes an adjunct therapy that then allows a better immune response to the tumor. Is that kind of the path you're going down?
Exactly. And there are actually, unfortunately, no good caspase inhibitors out there. There are clinically available caspase inhibitors, but they all get filtered out. And I'm trying to convince the pharma to go back into developing the next generation of caspase inhibitors. But this will be a game changer if we can do that.
And when it comes to the expected side effects of these caspase inhibitors, what would that be? What are you worried? Is there anything you're worried about, like if you bring this products to the clinic?
Sure, of course. I mean, side effects are, as always, in our minds. That's, of course, important. It's clear these are very powerful signaling cascades. You don't want to over boost those. But it's also equally important that most of these pathways that go into necroptosis, they're generally not expressed in our cells. So the pathways that involve necroptosis is a TNF receptor, but it requires also RIP3 and MKL. MKL is an interferon responsive gene. TLR3 is an interferon responsive gene. And another pathway that can activate necroptosis is one that is driven by CBP1. So this is nucleic zRNA-formed nucleic acid sensor, again, this is interferon responsive.
Mm-hmm.
So this pathway into necroptosis works best in an interferon responsive milieu.
Right.
And of course, when you think of these therapies, you need to think about how you can generate that milieu that is rich in TNF and interferon. And of course, you don't need to think far. Pathogens can do that, pathogen mimicry, of course, but radiotherapy does that locally. It drives production of TNF and interferon. CAR-T cells can do it, and immune checkpoint blockade can do it, because you have your incoming CD8 positive T cells that floods the tumor with TNF and interferon. So these scenarios indicate you can most likely have localized provision of the activation signal, while you then remove the brakes and provide also this caspase inhibitor. So the thing is also all these, the brakes to activate necroptosis, you will do that transiently. This is obviously not a treatment that you would give a patient all the time. You probably would also have to make sure that the patient doesn't have an infection.
Right.
Not that you have an overreaction.
Yeah, because I'm thinking, well, of course, I'm thinking of the gut and all the cells that are dying in your gut normally by, normal non-immunogenic apoptosis. But then if you're going to start messing up with that too, and you have, all the signals from the microbiome, and so the chances that you're, so I can imagine that that's probably one of the mucosal surfaces.
Yeah, and in fact, the gut is the most sensitive organ that we would need to be very careful with.
Mm-hmm. All right. I mean, there's clearly a lot of work ahead of you and your field, and I really hope that we can really harvest these pathways to improve the therapies that we have and stop, making tolerogenic chemotherapy treated cancer cells and start, using them to start up the immune response. Because I think in the end, it's clear that your immune response is probably the best tool you have against a tumor. You just need to give the immune system the right support and guidance. So I'm looking forward to see what's coming up next.
Absolutely.
So I think we're reaching the end of our conversation. It's been super interesting. Who would have thought with so many good things about death? And we like to ask our guest a question, to get them to know a little bit better, for our listeners to get them to know them a little bit better. So our question to you is, if there's any piece of advice you've ever been given, either, professional or not, that you would like to share with us today.
Yeah, there's a good piece of advice that I received, and that is believe in yourself. Try to do what you like best, even if it looks not very promising. When I started to study biology, this job which I'm doing didn't even exist, and biology at that time didn't really have a clear career path. But if you do what you like, you're more likely to do a good job. And if you do a good job, then you're more likely to not only enjoy life, but also end up somewhere where it's useful.
That's very nice. I like that piece of advice. You
Very
gotta believe in yourself.
That's how you end up in a poop company.
That's you, Jason. You end
Yeah.
up in the poop company. Yes. It's that I don't know if any of you have watched this TV series, Ted Lasso. It has the little the coach, and it has the sign on top of the door. It says "Believe." It sounds very, a little bit corny sometimes, but you know what? It is true. Sometimes I think it's important to keep that in mind. All right.
True.
So on that very high note, we're going to end our conversation. It was such a pleasure to have you. Thank you so much. We had Dr. Pascal Meier from the Institute of Cancer Research in London joining us for this episode of the Immunology Podcast. Thank you so much for your conversation with us.
Thank you very much, Brenda and Jason.
Thank you for being here.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest a guest.